Okay, so the second part of the lecture is going to be on just vision. So vision is obviously a more complex special sense, and it has quite a few pieces and parts to it. So I'm going to try to break it down as best as I can for you and make this, make sense without making it too complex as well. So believe it or not, about 70% of the body's sensory receptors are in the eye. That is how important our sense of vision is to us. And half of our cerebral cortex is involved, about half is involved in visual processing. Now, obviously, in an individual that especially was born blind, they will actually have a kind of an enhancement of their other senses, and those association areas also in the cerebral cortex that are associated with those other senses will also become more active. So, it's not to say that just because you don't have vision, you don't have good sensory perception, overall for your body. It's just if you, again, don't have vision, then you end up kind of developing kind of neural network rewiring. We talked about the plasticity of the brain in terms of those other senses. Okay. But anyway, so in most individuals, again, if you have vision, a large portion of your cerebral cortex is involved with visual processing. And while our eyes as humans can see many different wavelengths of light, there are other organisms that can see into the UV, infrared spectrum, as well as they can even perceive polarized light. Something, again, that we can't do. And so when you look at something like, because I teach marine biology, the stomatopod, which is the mantis shrimp, they way outgun us in terms of color vision. They actually have 16 different cones. We only have 3. So again, big difference between their color vision and ours. So what I want to do really quick is just review some of the basic structures of the eye. Hopefully you're getting most of these in lab as well. But obviously we have the eyebrow, which helps to provide, again, some protection from the sun, some shading. We have the eyelid here, also called the palpebra. So we'll call that the eyelid or palpebra. We have our eyelashes here, which are important, again, for things like our blinking reflex, stimulating that. This right here is going to be our iris, the colored portion. And then here's the sclera of our eye. Now, this whole outer layer here is actually called this portion here is called the sclera, and it's covered by the conjunctiva. And then we have the cornea here as well. And so this area here that's covered with the conjunctiva. And so basically right here, it'll tell you this is the site where the conjunctiva merges with the cornea. So the cornea here is going to cover the pupil as well as the iris. So that's the cornea. And then here the sclera is covered by what's called the conjunctiva, because as you're going to find out, the sclera actually is avascular. So the vascularization that you're seeing here in the white of your eye is actually due to this layer of conjunctiva that covers it. And that's usually the layer that kind of gets irritated if you get, an irritant in your eye. And then this area right here is called the palpebral fissure right here. There's the palpebral fissure. And then this area here is called the lacrimal caruncle. We'll talk about what that is, kind of an area where we collect tears. And then let's see, what else do we have? Oh, the pupil here in the center, and then the iris, the colored portion. And I think those are the main portions of the eye that you need to know. So again, you can review those for lab. I mainly am reviewing those for lab, and then I'll talk about the portions that are more important for lecture. So for example, when we talk about the eyelids, the reason that the eyelids are important to understand, or the palpebrae, if you want to call them, are that the eyelids blink reflexively every 3 to 7 seconds. You do it without even thinking about it, right? And the reason that you do that is it offers some protection from foreign objects and even things like dust that might get in your eyes, as well as it helps to spread secretions that keep that outer portion of your eye moist, where they keep it from drying out. Now, our eyelashes here have follicles that actually have some nerve innervation, and those nerve endings are what initiates your reflex, your blink reflex, when something kind of brushes your eyelashes. That's just a protective reflex, obviously, again, to protect your eye. If something's that close to your eye that it's rubbing up against your eyelashes, then your eye will reflexively close to protect your eye. We do have these lubricating sebaceous glands that are associated with our eyelids. One of the big ones is called the tarsal or meibomian glands. The tarsal or the meibomian glands are these, again, the big modified sebaceous glands that are going to produce oils. And that oily secretion, again, will help to kind of lubricate the lid and the eye. It's also what keeps your eyelids from sticking together. So that lubrication prevents them from sticking together. So there are two kind of dysfunctions that you can have with these, or not disorders, but like, temporary issues that you can have with these meibomian glands, or tarsal, probably more commonly called the tarsal glands. One is a chalazion. Chalazion is where that tarsal gland becomes obstructed and blocked, and then it builds up this kind of, large kind of inflamed bump on your eyelid. So that's called a chalazion. Now, if you get one of these sebaceous glands that is blocked, not necessarily on top of your eyelid, but one that is right on the edge of the eyelid. Eyelid. If it's right on the edge of the eyelid, we call it a stye. And that usually happens just when you have some inflammation of those sebaceous glands, and it kind of partially blocks it. But it's usually not quite as big as the chalazion, which is the kind of a fully obstructed tarsal gland. So this one's just kind of some inflammation, caused kind of a little bit of a blockage. And it's just right along the eyelid. So that's what we call right along the edge of the eyelid. That's what we call a stye versus the chalazion. So you can see those two here. Here's a picture of a stye. And then I just put those because you might see those. Sometimes people even come into the doctor's office with them because they're not sure what they are. Ciliary glands we have these ciliary glands between hair follicles, and they're basically just these like modified sweat glands that we have. And again, I'm not going to talk too much about them because they're not as important for our purposes. So here you can see some interesting things. If you look here, I like this diagram because it actually shows you the conjunctiva. I don't care that you know the bulbar conjunctiva, which is this portion, versus the conjunctival sac, and the palpebral, which is this area here, conjunctiva. I just want you to know that this is all conjunctiva that covers, underneath the eyelid all the way through up to what's called the cornea. The cornea is going to be this clear portion here. So it'll cover the sclera. And again, as we're going to talk about in just a minute, the sclera, or the white of the eye, is avascular underneath. So it's really just this conjunctiva layer that is vascular and actually has, it gives us our red eyes. Like if you didn't sleep and, someone says, oh, your eyes look red, it's actually the conjunctiva that's irritated. So kind of interesting. And again, you can go over kind of all of the other structures again, but it's also nice because it shows you that here a lot of these extrinsic eye muscles are attaching here to the sclera, you can see. The lacrimal apparatus is obviously going to produce our lacrimal fluids, which are tears. And so this is going to consist of our lacrimal gland. And like any gland, it's what's going to make the tears. And then we have ducts that are going to go from the gland and drain, again, onto the eye, and then eventually drain it off the eye into the nasal cavity. And so we'll talk about how that happens. And the lacrimal glands, I'm just going to kind of go up here. Here's the lacrimal glands up here. Again, they're going to produce the tears along with some other secretions. So we call them lacrimal secretions because there's other things in those tears besides just kind of like a saline kind of solution. It also contains some mucus that has antibodies in it to provide an immune function, as well as antibacterial lysozymes. Okay, so this helps to protect, to clean, lubricate, and moisten our eyes. Now, the challenge is that we don't have a ton of immune function here, in that we basically are relying on our mucous membranes here, along with some antibacterial lysozyme. But this is actually a good place for things like viruses to enter the body. So our eyes and our nose are primarily protected by mucous membranes. And the challenge here is that we don't have the protections that we have in other areas of the body. You know, a lot of people are always worried about, you know, taking in things by mouth, but a lot of times, you know, you have stomach acid and things like that that will kill off some bacteria. I always say, again, it's about bacteria numbers, but it kills off some bacteria and some viruses. But our mucous membranes and our nose and our eyes are really vulnerable areas. So if you want to try to like prevent yourself from getting sick, one, wash your hands, but two, also really try not to rub your eyes or your nose. It's hard to do sometimes, but really think about trying not to rub your eyes and nose. Because again, we just have these mucous membranes and, you know, some antibacterial enzymes, but you know, that's about it. Now, obviously blinking is going to spread the tears, and it's going to push them towards that, what we call the lacrimal caruncle. Which is in the, I like the lacrimal caruncle term because it's just funny. Like it's kind of spelled like car uncle, but it's caruncle. And so that's just the corner of your eye. I'm not gonna have you know medial commissure and lateral commissure. It just seems like more, you already have commissures to memorize with the brain. It just gets to be too much terminology. So lacrimal caruncle is fine. So again, as you produce, oops, sorry, I went the wrong way. As you produce tears from those lacrimal glands, they're going to move down these lacrimal ducts onto the surface of the eye, and then they'll be pushed across toward this lacrimal caruncle, okay? And then when they collect in this lacrimal caruncle, it's oftentimes where you also get some of the oily secretions that form, like some people call it like the sleep in your eye. It will build up in that lacrimal caruncle. But the tears, okay, for the most part, your normal tears, unless you have excessive tears, the normal tears will actually drain through this hole called the lacrimal punctum. It's kind of, I know, funny names, right? Lacrimal punctum. And that's going to drain your tears off. So lacrimal puncta. So again, you don't have to know medial commissure. You don't have to know lacrimal canaliculi. It's too much, right? I know you guys are feeling that way. So lacrimal caruncle. To, if you want to write down how this goes, it goes lacrimal glands to the lacrimal ducts, across the eye to the lacrimal caruncle, and then drains through the lacrimal punctum. When it drains this lacrimal punctum, it's going to go from the punctum through these little canals into the nasolacrimal duct, okay? And so this nasolacrimal duct is draining into the nasal cavity. So this is why when you do, cry or your eyes are irritated for some reason, they're producing excessive tears. This is why your nose runs. It's because, again, you have more tears draining into that nasal cavity through that nasolacrimal duct. Okay, so it's kind of important to understand that nasolacrimal duct. You can also tell your kids, if they're crying sometimes and then they get mad because their nose is running and they're mad at you because that happened. You can tell them, it's not your fault because when they cry, they make their nose run. It's just it has to go hand in hand. So, that's a fun one always to share with your kids. Not that in the moment that it probably is, they probably are frustrated with you in the moment, but maybe later on you share it with them. Now, of course, again, this is what I was just talking about. When lacrimal secretions increase, because maybe, again, you've irritated your eyes, or maybe you've had an emotional response that has caused, again, you to produce more tears, then those tears will spill over the eyelids and fill the nasal cavities. And that causes, again, your tearing as they spill over the eyelids, as well as the sniffles, because, again, it's draining through that nasolacrimal duct. Okay. So I put some of these here about, again, cutting onions. Again, that does release chemicals that can irritate the eye and cause your eyes to tear more excessively. Or maybe you're having an emotional response, like for me, the fact that The Office was leaving Netflix. That was, an emotional moment for me. So as you guys know, I like Office. The Office is one of my favorites. But okay, so I digress. We're going to get on to the extraocular muscles. So as you probably well know for lab now, we have these extraocular muscles that we primarily call the rectus muscles. We do have some oblique muscles in there, like the superior oblique, but so these are what we call our extrinsic. And I think for lab, I think you just, actually, I get you guys mixed up with 160. I don't know if you have to just know the extrinsic eye muscles, or you do have to know, I think you do have to know that there's the superior, here's the superior rectus muscle, but they're pretty easy, like superior rectus muscle, inferior rectus muscle, lateral rectus muscles. And so these are all of the, basically all of the muscles that are going to move your eyes, okay? And they're innervated by the different cranial nerves. We'll kind of review them. So in addition to these extrinsic eye muscles, the eye also contains some intrinsic eye muscles that we'll talk about called the ciliary body and the iris. They're going to be the two muscles that we're going to talk about inside the eye. Ciliary body is really important for helping you with your accommodation for near and far vision, because it's going to influence the shape of the lens. And then the iris will influence the shape of the pupil, and that will determine how much light enters the eye. So those are really two really important intrinsic muscles that you will have to know. And they're actually controlled by the autonomic nervous system. Both of them are controlled by the autonomic nervous system. So again, I'm not going to have you know the cranial nerves and which specific muscle is associated with them. Again, you just have to know that for cranial nerve 3, the oculomotor one, it's going to be involved with, again, moving the eyes up and down as well as medially. The trochlear nerve is going to be associated more with kind of, remember, rotating, turning that eye. So it says depresses and turn it laterally, but it kind of rotates it. And then the abducens is going to be associated with moving the eyes laterally. So that's going to be, especially like if you're moving your eye side to side. I always say that's the sus, because my kids say for suspicious, sus. That's your sus cranial nerve, your sus muscle here. Okay. So sometimes, again, if these external eye muscles from one eye to the other side, like your left and your right, external eye muscles aren't perfectly coordinated, this is what can cause double vision. And so obviously if they're not coordinated together, then you can't properly focus the image on the same area of that same area, the visual field, from each eye. So you end up getting two images instead of one. And this can happen sometimes if you have paralysis or weakness of a certain extrinsic eye muscle, can cause kind of like a lazy eye, or neurological disorders can also cause this. So sometimes if you haven't had this and all of a sudden you develop it later on, then it could be a potential, indication that you've got a neurological disorder. Again, not always, just again, sometimes. So there is something called strabismus, and this is a congenital weakness of the external eye muscles that you're born with, in which the affected eye either rotates medially or laterally. And if this isn't taken care of when you're, a young child, if you don't correct this early on, what can end up happening is the brain actually stops listening to this kind of dysfunctional eye that's giving kind of the mixed messages, and it can actually result in functional blindness in that eye because the brain just stops listening to it. So it's kind of interesting. So that's why if you have a child that's born with strabismus, then you'll have to they'll definitely make efforts to correct that early on. Okay, so now what we're going to do is we're going to talk about the structure of the eye and the different layers of the eye, because there are these three major layers that we're going to break up the eye into. So the first layer is going to be called the fibrous layer, and it's going to consist of the sclera, which is going to be the white of the eye here, and then it's going to be the cornea, which is going to be the clear section right here. So it's basically this kind of if you think of it like the inner and outermost layer. It's kind of hard to think of like an inner outer layer, because out here it's the outermost layer, but down here, it would be the kind of innermost layer. So it's just this, whatever you want to think of, an outer layer on the outside here. So again, cornea, and then this runs into the sclera here. Okay. That's the fibrous layer. Now the next layer in from that is going to be the vascular layer, and it's going to consist of this choroid layer that's going to have a lot of blood vessels. Then it's going to consist of the ciliary body, which we're going to talk about right here. That's going to attach to the lens of the eye through these little ligaments. And then it will also include the iris, which you can see it's actually continuous with the iris right here, which is that muscle that gives you the color of your eyes. If you have blue eyes, it's because your iris has that pigment. And then you're going to have the innermost layer right here. And this innermost layer, which they kind of drew in this like yellow here, is going to be the retina. It's the neural layer. And so that's going to be the layer that has, and here they do the retina right there. That's going to be the layer that has all of the receptors for your sense of vision. So the rods and all the cones you're going to find in that layer. Now we also have these internal cavities that are filled with fluids, and they serve many important functions that we're going to talk about. But as you can guess, maintaining the shape of the eye is a big one. And so we call these humors. So you'll see we'll talk about the aqueous humor up here, and then back here we'll talk about the vitreous humor back here. This is fun when you dissect, because the vitreous humor is like jelly-like, it's gelatinous-like, so it's kind of cool when you take that out. And then the lens actually is going to separate the cavities into an anterior cavity. So from the lens on up forward is the anterior cavity. And again, this will be filled with what's called the aqueous humor. It'll be a different liquid. And then from the lens all the way back here is going to be the posterior segment or posterior cavity. And this posterior cavity is going to be filled with vitreous humor. So we'll kind of talk about that. And so it shows it here, kind of posterior segment is going to be filled with vitreous humor, and then anterior segment will be aqueous humor. Okay, or anterior cavity. You can call it either anterior cavity or anterior segment. This book calls it anterior segment. If you have the Tora, it'll call it anterior cavity. So I'll try to make sure I put both on an exam. Now, again, our first layer that we're talking about is going to be this fibrous layer that we're going to talk about in more detail here. So remember that this fibrous, oops, sorry, I just skipped ahead there. So remember that this fibrous layer is going to be this, again, kind of outer layer here that consists of the cornea, and then it's going to run into the sclera here, okay? So let's first talk about the sclera. So the sclera is going to be this portion back here. It's going to be this kind of opaque posterior region. It's going to run all the way back here to the optic nerve. And it's going to help protect the shape of the eye. So it's kind of a fibrous, dense connective tissue. Also, again, important to remember that this is avascular. So both the sclera and the cornea are avascular, okay? This is also going to be where the extrinsic eye muscles are going to attach. They're going to attach to the sclera there, okay, to be able to move the eye. Now, at the junction right here of the sclera here and the cornea, there is going to be a tiny hole. It'll be right here, again, at this junction of the sclera and the cornea. And there's going to be a tiny hole called the scleral venous sinus. Let me see if I can find it in a diagram here. Here it is. So here again is the sclera. Here's the cornea right here. And here's this little opening called the scleral venous sinus. It's going to be really important for draining off the aqueous humor from this anterior cavity. So I just like to mention it here, and then I'll mention it again later, but that way then you're kind of getting it twice. And then obviously posteriorly here in the back, you're going to see this is where the optic nerve exits the eye. So this is also called the optic disc on the retina, or the blind spot. So this would be where your blind spot is. So this is, again, optic nerve or cranial nerve 2 is exiting here with, again, carrying back all that information from these sensory receptors in the eye. And we'll talk about how it does that. And then, I don't really care that you know this too much. This is getting kind of nitpicky, but the sclera is continuous with the dura mater of the brain as it goes in. But again, not really super critical that you know that. Cornea. So the cornea of our eye here is where we put our contact lenses on top of, right? To help kind of reshape that cornea if it's like mine and misshaped, and you end up being myopic. So that cornea, again, is transparent. It's, you know, just this anterior layer here. And it forms that clear window that's going to allow light into the eye. We'll talk about how it also is a refractive layer. So as light enters, it's going to bend the light, and then it will bend again when it hits that lens. So that cornea is an important refractive layer. It also helps to protect us from, you know, abrasions and things like that. Although it's still quite painful if you ever have scratched your cornea. Again, that inner layer will, cornea endothelium will help to try to maintain some clarity of the cornea as well. And there's an epithelial layer that's on the outside and the inside surface. Again, you know, this is really getting kind of detailed. Your book gets pretty detailed. I'm not going to test you on the epithelium that covers the cornea. I want you to know the big things. That the cornea forms this clear window, lets light enter the eye, and that it's going to refract the light as well, which we're going to talk about in a few minutes as that light enters the eye, okay? There are, again, numerous pain receptors, as I just talked about, and that helps to contribute to blinking as well as our tearing reflexes. And so this is why some people actually can't tolerate contact lenses. Their pain receptors just don't ever seem to adapt And so they just can't tolerate contact lenses. Okay, so corneal transplants, because the cornea has no blood vessels, remember it's avascular, just like the sclera, it's beyond the reach of the immune system. And therefore, the cornea is the only tissue in the body that we can safely transplant it with very little risk of the person that is rejecting that transplant. So corneal transplants are pretty amazing and usually pretty successful. So that's our outer layer. The sclera and the cornea are that outer fibrous layer. Okay, so now let's talk about the vascular layer, which is one layer in. Okay, so the vascular layer is in pink here. And this is going to consist of what's called the choroid, which is, again, carrying a lot of blood vessels here. And then here is going to be the ciliary body. And then here's the iris. Okay, we'll talk about what each of these do. So the choroid region is going to be from here on back. Okay, here's the choroid region. And it's this posterior portion. Okay, it's going to supply blood to all the layers of the eye. Okay, so it's going to provide the blood flow and the nutrients and things like that are keeping things like the sclera going, as well as the retina. So that's why it's in the middle. The choroid has to be in the middle because, again, it's supplying the nutrients and things that are needed for both sides, the sclera and then the retina right inside. So hopefully that'll help you remember that. And then there is a brown pigment to it. It actually helps to absorb light to prevent scattering of the light as it enters the eye here. So again, that typically, if there was a lot of scattering of light in there, that would cause some kind of visual confusion inside the eye as it's like bouncing around, stimulating all these receptors. So that brown pigment is actually produced by melanocytes, is actually a very important function of the choroid to prevent scattering of light. Okay. Now the ciliary body is right here, this section right here, and it's going to attach to these little what we call suspensory ligaments that attach to the lens of the eye. So here's the lens of the eye. And really quick, I'm going to kind of jump ahead for 1 second and talk a little bit about what the lens of the eye does, even though we haven't gotten to the lens yet. But the lens of the eye is going to be this kind of very important crystalline structure. Kind of looks, when you cut it in half, like the layers of an onion. And the lens of the eye can stretch, and then it has some elasticity to it, and it will bounce back. Okay, so this is going to be really important for changing the way that the light is refracted and enters the eye for when we do something called accommodation. Okay, so for example, if you were looking at this lecture and you are close to your computer screen, okay, what's happening is your lens of your eye is adjusting to the kind of close distance that you have to your computer screen. And what's happening is the lens actually kind of relaxes a bit as you're looking at your computer screen. Now, what ends up happening is as you move farther back and you look at something far away from you, when you do that, the lens of the eye is actually going to be stretched out. So again, because of these, we'll talk about how that happens with these ciliary body muscles here. But it's actually going to stretch out the lens, and that changes the way the light enters the eye and allows you to look far away. Now, to help you remember this, what the lens does, whether you're looking up close or far away, is I'm going to tell you what happens to us as we get older. As we get older, that lens of the eye loses some of its elasticity, because think about it, it has a lifetime of being stretched and then bounce back, stretch and bounce back, stretch, bounce back, stretch. And so what ends up happening is it loses its elasticity. So if you're like me, and I've hit this point where I'm starting to see this, your lens has lost a little elasticity. And so when you go to read something up close, your lens doesn't have the capability of bouncing back all the way after it had been stretched out looking far away. So it doesn't bounce back all the way, and you have trouble reading up close. And so then you need to kind of like take the book and like move it back a little bit. And that's because your lens has lost that elasticity, that ability to bounce back all the way. So that's kind of what happened. That's basically what's happening. When you look far away, the lens is stretched. When you look up close, it bounces back. Okay? So these ciliary muscles that are here in the ciliary body, that's one of the main functions of the ciliary body, is that it contains these ciliary muscles that are going to control the shape of the lens. And they do that by being attached to the lens through these suspensory ligaments. Now, I know your book uses ciliary zonule, but I just have always taught it as suspensory ligaments. They just recently changed this. And so because you're more likely to hear suspensory ligaments, because that's what it was, I'm going to use suspensory ligaments. So again, these ciliary muscles are going to be attached to suspensory ligaments that are attached to the lens. And that's how they'll be able to hold the lens in place and stretch it out and allow it to relax and bounce back when you're doing what's called accommodation, looking near and far, near and far. So those are the main functions of the ciliary body. Here's a close-up so you can see again these ciliary muscles here, and then you can see they're attached to the lens through these suspensory ligaments. You can see how, again, during contraction and relaxation, that will stretch out the lens and then allow it to bounce back. And again, we'll talk about how that does that. Okay. So again, the ciliary muscle controls that tension on the ligaments and hence the lens, and allows us to do accommodation near versus far. Okay. So hopefully that helps out a little. And then here just is showing you some of these structures. It's pretty cool to see some of these structures on an actual dissected eye. So you can kind of look at that. Now, when we are talking about the vascular layer, we have talked about the choroid and we've talked about the ciliary body, but we haven't talked about the iris yet. So the iris is the third part of the vascular layer. Obviously, this is the colored part of the eye that's going to lie between the cornea and the lens. Okay, so here you can see right here, here's the iris, and it lies between, here's the cornea, and here's the lens, smack dab in the middle there, okay? And it is continuous with the ciliary body because, again, it's part of that vascular layer. The pupil is the portion where there's actually no iris, okay? So the pupil, it's kind of funny because we think of the pupil as a structure, but the pupil is actually not. The pupil is actually nothing. It's just where we don't have iris, okay? So it's going to be that opening that's going to be important because it regulates the amount of light entering the eye. So when we are looking at something up close or we have bright lights, this is actually going to cause one of these two muscles in the eyes to constrict. And so, sorry, I haven't talked about these two muscles yet, I just realized. So we have two different muscles. One of the muscles is under parasympathetic control. It's the inner one. And then the outer muscle, called the radial muscle or the dilator pupae, is under sympathetic control. So let's talk about this first muscle that's called either the sphincter pupillae. I kind of like that name because it is like a sphincter. It's circular. And so as it contracts, it's going to close this opening here, just like a sphincter, and make that pupil smaller. So it makes the pupil constrict. So our sphincter pupillae constricts the pupil, and this is a parasympathetic response, okay? So this parasympathetic response happens when we're looking at something up close, or you put a bright light on someone. If you wanted to check for the pupillary response in someone, you would take a flashlight and put it across their eye really quick, and you should see this muscle here, the sphincter pupillae, or again, you can call it the circular muscle, contract and that pupil constrict, okay? Now, the opposite would happen for if you were under sympathetic control. So again, let's say, you know, the scary bear is chasing you. Then what's going to happen is this other set of muscle called the dilator pupillae, because it dilates the pupil. So it's kind of an easier term, dilator pupillae. Or you can remember radial muscle, because the muscle radiates out from the center here. And that's exactly what it's going to do. As it contracts, it's going to pull this inner sphincter pupillae muscle outward. And as it does that, it's going to basically dilate the pupil, okay? So, and remember that happens with the sympathetic nervous system control, because, you know, you want to be able to see the scary bear that's chasing you, even if it's at night. And so you're going to allow more light in, okay? And this is also why, like, if someone's lying, a lot of times their pupils will dilate, because it stimulates the sympathetic nervous system. So again, changes in emotional state can cause the pupils to dilate. Also, when a subject matter is appealing, it can cause your pupils to dilate. Or when you're really like heavily in thought process for, you know, problem solving, doing some critical thinking, then your pupils can also dilate as well. So kind of interesting. So again, this is just showing you, you know, parasympathetic, your rest and digest. Your sphincter pupillae is going to contract. Here, you know, sympathetic nervous system. Here, this guy literally was mountain biking and he was chased by a grizzly bear. So clearly, his dilator pupillae was like an overdrive, right? Allowing as much light in as possible so he could maneuver his mountain bike out of the way of the grizzly bear. So again, maybe those stories will help you remember those two things. And hopefully the fact that you have the autonomic nervous system along with this, it'll help you kind of remember that little overlap there. Okay, so this inner layer is going to be the last layer that we're going to talk about. Sorry, I got to drink water there. And this inner layer is going to be the neural layer. This is going to be the layer that's going to be filled with the sensory receptors for your vision. So this is going to be called the retina, okay, that inner layer. And the retina originates as kind of an outpocketing of the brain. So it contains, again, lots of these photoreceptors and neurons. So it contains millions of photoreceptor cells that are going to transduce light energy. They're basically, all this means is they're going to convert the light energy into nervous conduction that our brain can process, okay? So there are also other neurons that are going to be involved in this processing that we'll talk about that are going to be important for the overall process. And then we also have glial cells. Those glial cells are just like, again, when we talked about neuroglial cells in the brain, they're going to be your support cells, okay? So there is this kind of dedicated two-layer membrane. There's an outer pigmented layer of the retina, and then the inner neural layer of the retina. And that inner neural layer is the only layer that plays a direct role in our vision. So that inner neural layer is just like it sounds. It's got the photoreceptors and the neurons. So here, excuse me for that, here is the retina, okay? And this is the neural layer of the retina. And then here, this layer right here is going to be the pigmented layer of the retina, okay? And so we're not going to say much about this pigmented layer of the retina. It's just this little pink layer right here, really thin layer, just a single cell layer thick. It's right up against the choroid, which again, the choroid is that layer, that's the vascular layer. And that's kind of makes up the middle layer. And its main function is to absorb light. That's why it's pigmented, and prevent it from scattering. So again, if light were like bouncing back and forth and scattering, as the light enters here and activates some of these photoreceptors, we don't want it bouncing all across the eye, because again, then it's going to give us these like mixed signals. So As light enters here and stimulates these photoreceptors, then in the neural layer, then it will be absorbed in that pigmented layer. Okay, so that's its main function. It also kind of will phagocytize or eat up some of these old photoreceptors as they break down. And then it also will store vitamin A. We're going to talk about why vitamin A is important in just a minute. So this neural layer of the retina is where we're going to spend the rest of our time for this lecture. And this neural layer has these this like serrated edge called the ora serrata. Let me see if I can find where the ora serrata, like a picture. I should have put a picture near here so that you could see the ora serrata. But it's basically, oh, here it is. Here's the ora serrata. So here, the ora serrata here is just going to be right here where the retina is going to end here. And so where that retina ends and kind of meets up with this like ciliary layer is going to be what we call the ora serrata. Okay, and so you can see it's called ora serrata because it's serrated. And you'll see that when you look at the dissected eyes. So again, not a super important, I'm not going to test you on that, but I show it to you so you can, you know it for lab. But the main thing that I want you to know about this inner layer of the retina is that it's what contains the photoreceptors that are then going to stimulate these bipolar neuron cells, or bipolar cells. They're not technically neurons, bipolar cells. And then those are going to stimulate the ganglion cells. So what is going to happen is it's going to look somewhat like this. So you're going to have your receptors, your photoreceptors. So here again is this neural layer of the retina right here. You're going to have your photoreceptors, your cones and rods here, and they're going to then, again, after they're stimulated, pass on that message to a bipolar cell, which will then pass on that message to a ganglion cell. So there's these kind of three layers in the pathway. And then it's these ganglion cell axons that are going to make up the optic nerve. They'll go back down here and make up the optic nerve. So that's basically how our signal is spreading. It's spreading from photoreceptor to bipolar cell to ganglion cell. And again, those ganglion cell axons are what's going to make up that optic nerve. Okay? Now, we do have one challenge in that in our retina here, in our neural retina, there is going to be one area where there are no receptors. If you see, like the receptors ended here and here. And so because this area lacks receptors, it's what we call our blind spot or the optic disc in anatomy. So in anatomy, in lab, you're going to call this the optic disc. But in the general world, we call this your blind spot. And so the crazy part is every day your brain plays tricks on you because this blind spot is there all the time. So why don't you see it? The reason you don't see it is that your brain fills in the gaps for you. How crazy is that? So there are all kinds, you can click on this link here, but there are all kinds of things that you can do to test this. This one's kind of fun and easy. You can even do it on this slide. You're going to cover your right eye, take your left eye and focus it on this plus sign. And then you're going to move yourself closer and closer while your left eye focuses on that plus sign. And eventually you're going to hit a point where this hole disappears. And when it disappears, you've hit your blind. The light that's entering from there, this point has hit your blind spot on that eye. And your brain is basically filling in the gaps as best as it can. And it fills in these black lines for you. It's wild, it's wacky to do. So if you have a little time, go to this site and they have a ton of these exercises like this to test your blind spot and how your brain fills in the gaps for you. And it's just fascinating. So please, if you have the chance, take the time to do that. Now, when you look at the retina, obviously you have many photoreceptors, so 250 million photoreceptors. Again, I'm not going to test you on the exact number. It's a lot, right? And so you're going to have two major types of photoreceptors. This you do have to know, and you probably should know this already. You have your rods, and then you have your cones. And it's easy to remember. Cones start with C. That's for your color vision. So your rods are going to be for your night vision. Okay, so think C, cone, color vision. And you actually, we'll talk about the cones, I guess, in just a minute. I forgot I had rods here first. So your rods are going to be for, again, a situation where you have dim light, or you're using, again, your peripheral vision receptors. You're going to see you have a lot of rods in the kind of periphery of your eye. These are going to be more numerous and more sensitive to light than your cones. So just a little bit of light will trigger them off, where cones need a lot of light to trigger them. Okay. So our rods, again, are no color vision, but they do no color vision and then no sharp images. But again, they are receptive to dim light. So that's the big thing. So no color vision, no sharp images. That's all your cones. Okay. But our rods are the receptor that we have in the greatest number in the periphery. Okay, so again, dim light, peripheral vision. Those are the two that you got to remember: dim light, peripheral vision. And so again, this is just showing you that pathway. When we activate our rods with that dim light, as the light enters here and it activates that rod, then that rod will then stimulate a bipolar cell, which is then going to stimulate the ganglion cells, and then that will send that message back through the optic nerve. So again, we have rods and then we have cones. Cones are obviously going to be associated with our color vision. You probably already know that. This is our vision receptor for bright lights. It's also going to give us our high resolution color vision. And it's also going to give us our clearest images. And that's because we have this area called the macula lutea. The macula lutea is going to be this area posterior and kind of lateral to the blind spot. And this is an area where we have a really high density of cones. And then if you move to the center here, called the fovea centralis, the fovea centralis is this tiny pit in the center of the macula lutea. And this is the area that contains all cones. And this is our area of highest visual acuity. So what this means is when you're reading these words, fovea centralis, okay, as you read that word, fovea, you are directing the light into the eye from this area, fovea, onto your fovea centralis, okay? And so the reason that you move your eyes as you're reading left to right, the reason your eyes will move is that you're going to keep focusing that light as you move from here to here onto the fovea centralis. So you have to move your eye. And that's because, again, that fovea centralis is that region of highest visual acuity. Okay, you're going to get the clearest image there. So again, our eye movements allow us to focus in on an object so that that light can be picked up on that fovea centralis. Now, what's interesting is when you look at something like an eagle, eagles have a even higher density of cones than we do on that fovea centralis. And that is why their visual acuity is better than ours. So kind of interesting. So again, this is just kind of showing you here this macula lutea region. In the very center, you can't really see here, but in the very center would be the fovea centralis where you have the highest density of cones there. This is also showing you the optic disc. And then also going to show you some things that we're going to talk about, like the central artery and vein that are going to emerge from the optic disc. This is kind of cool because this actually shows you your pigmented layer of the retina here. And then this is your neural layer of the retina. And then it shows you the different receptors and then the cells of them. So here you can see these are the nuclei of your photoreceptors, your rods and cones. Here then, that message would be passed on to your bipolar cells. So there's the nuclei of the bipolar cells, which are then passed on to your ganglion cells. So kind of cool to see those layers there. Now again, this innermost layer does have, again, some two sources of blood supply. And so one source is, of course, that choroid layer that's just to the outside of the retina. And it's providing, again, nutrients and things through blood supply through that choroid layer. And then we also have the central artery and vein. The central artery and vein are coming in here through, you can see the central vein. Here's the central artery coming in through the optic disc. Okay. So those are just the main sources of blood supply. You don't have to know which one's the outer third. I just want you to know that there's the two, choroid and the central artery and vein. And that's it. And it supplies, again, the retina with the blood supply it needs. When we look at the fluids, okay, in the eye, so now we're going to talk about the kind of internal chambers and fluids. We have what's called, again, the anterior cavity or anterior segment. And then we have the posterior cavity or posterior segment back here, okay? And the lens is the determining factor for anterior versus posterior. So the lens is that dividing point. So the posterior segment behind the lens is going to be filled with this cool stuff called vitreous humor. The vitreous humor is going to be gelatinous, like Jell-O. So if you do the dissection, you can feel it. And it's going to be important for, again, transmitting light, because that light is going to enter the pupil, have to move through the vitreous humor to the retina behind it. Also, one of its main functions is to help provide shape of the eye, to help provide the shape of the eye and hold the retina firmly against the pigmented layer. So it's going to hold the neural layer of the retina against the pigmented layer of the retina. So it really is providing the pressure for holding the shape of the eye in the back there. So it contributes that what we call intraocular pressure, the pressure inside the eye. And so what's interesting is that vitreous humor, as we get older, we can end up with debris in that vitreous humor, and it shows up as these floaters. I don't know if some of you have these. I have some. And so, yeah, it looks just like this. Some of them look like this, and some of them look like this. And a lot of the times, again, we, because of, our sensory adaptation, we kind of ignore them. But sometimes if you're looking at a flat white board, then you really notice them. You're like, oh, I didn't realize they had those. But then again, give it 5 minutes and you'll adapt and stop paying attention to that. Your brain basically stops paying attention. So when we look at we had the eye again, we had the posterior cavity or posterior segment, which was back here. And then we have the anterior segment, which is the lens forward. So again, posterior with a vitreous humor is lens back. That's all vitreous humor back here. And then here, lens forward is going to be aqueous humor in terms of the cavity itself. So when we look at this anterior segment, we can divide this anterior cavity into another anterior chamber and posterior chamber. I'm not going to ask you about this for lecture. It is in your lab, so I'm just going to show it to you. So when we look at this anterior cavity, this is going to be the anterior chamber from the iris forward here, the anterior chamber here of the anterior cavity, and here's the posterior chamber back here of the anterior cavity. So what's going to happen is the eye will make the aqueous humor. So the vitreous humor is a little bit different because it's encased in a sac, but here the aqueous humor is continually made And so again, unlike the vitreous humor, it's going to be continually made, and it's not held in a sac, and it's going to flow through. And we already talked about it's going to drain at this, what we call the scleral venous sinus. I'll show you this in just a minute. It's right at the junction of the sclera and the cornea. It's a little hole. Or you can call this the canal of Schlemm, okay? And so this is important, this flow of aqueous humor, because the aqueous humor is going to supply nutrients and oxygen to things like the lens that can't get it any other way, or the cornea of the eye that can't get it any other way. And then also help to remove wastes as well. So basically what's going to happen is that it's super easy. The aqueous humor is continually going to be produced by some of these capillaries in the ciliary body called the ciliary processes. So these ciliary processes are going to secrete the aqueous humor. It's going to flow up here in that posterior chamber up to the anterior chamber, and then down the that scleral venous sinus, or the canal of Schlemm. I kind of like the canal of Schlemm because it's named after the scientist that discovered it. And so it would be drained off here and then into the venous system. So you're continually producing new and draining the old, okay? And that also helps to keep that fluid clear and free of any debris. Now, glaucoma is a condition in which the drainage of that aqueous humor is blocked for some reason. So for some reason, this scleral venous sinus isn't properly draining, and you end up, as you can imagine, building up aqueous humor and pressure inside here, right? And so as that pressure builds up within the eye, it can increase to some dangerous levels that would eventually cause the eye to compress on the retina as well as the optic nerve. And if you let it go too long, it can actually lead to blindness, okay? Because as you can see, it would just continue, as you build up pressure in here, it's going to put pressure back here and build up that intraocular pressure. So there are a few kind of early warning signs, but again, glaucoma is one that it's a little tough to notice until it's kind of really, really not bad, but really bothering you. So that's why it's good to go, see your eye doctor and be tested every once in a while for glaucoma. So there's a few earlier signs, but some of the later signs are going to include things like halos around lights, as well as, blurred vision that just doesn't seem to be corrected with your glasses. You're always kind of having to change your prescription. So those are some signs that you could have, again, the beginning stages of glaucoma. Let's talk about this really important lens that we already talked about. This lens is a biconvex, kind of transparent, flexible material. It changes shape, as we talked about. And I'm not going to talk about the lens epithelium versus lens fibers. But what I do want you to know is that lens does have this kind of crystalline structure, this protein crystalline in it. And that protein crystalline is kind of like wrapped around itself in like an onion, like in layers, like an onion. And so that lens, again, is going to be what's called convex. And remember we talked about it's going to stretch when you need to see something far away, and then it's going to bounce back to its original convex shape when you're looking at something up close. Now, as we get older, that lens is going to lose its elasticity, and it's going to end up making it more difficult for us to read things up close or for near vision. So we call this presbyopia. And we all are likely to end up with that at some point in time. Now, when you talk about cataracts are actually clouding of the lens. So again, glaucoma is a buildup of aqueous humor in the eye, but cataracts are clouding of the lens. And this oftentimes causes the world to kind of appear distorted It kind of makes it look like we're looking through frosted glass. So when your cataracts are bad enough that you really, need to regain your vision, they can actually go in and replace the lens with an artificial one that isn't so clouded. Really quick, we're going to talk about the optics of light as it enters the eye, and then we'll talk about how that stimulates different receptors. So remember that when we're looking at the wavelength of light that is going to be used by our eyes, it's going to be in this little small section here called the visible light range. And so the visible light range will be in between UV and infrared here. And that visible light is going to have wavelengths in between 400 nanometers and 700 nanometers. And so our eyes will only respond to that visible light. We're not able to see into the infrared and the UV. So we do have different receptors that are going to be stimulated by different wavelengths of light. So we have our blue cones here that are going to be stimulated by 420 nanometer wavelengths. We have our rods, which are going to be stimulated by 500 nanometer wavelengths, and again, around that area, it's not just by 500 nanometer wavelengths. And then green would be around the 530 nanometer, which is your green range. And then red, which would be, again, 560 nanometer wavelengths, which would be kind of in that orange range on up. So, those are your different receptors, and the different receptors, again, will be activated by the different wavelengths of light. So we'll talk about how that is. So yeah, and you can see your rods are going to be kind of if you look at your rod, they end up, sorry, I think I said this is the blue range, but they kind of end up being in this range that's more of that kind of dark blue night range. So again, we have these kind of darker wavelengths that are associated with that. So let's look at wavelength and color. So when we talk about light, just remember that light is these packets of energy called photons, but they travel in a wave-like fashion. And some of them are going to have these longer wavelengths. So they're going to go up and have these long, let me see if I can find something that shows the wavelengths. I don't think I do have anything, but anyway, some of them will have these kind of, if you follow my pointer here, longer wavelengths, okay? So something like this. And so that's going to be closer to like your red wavelengths when you're talking about the spectrum, okay? They have a longer wavelength of light. And that's why, again, they're going to be closer to 700 nanometers because they have that longer wavelength. Our shorter wavelengths of light are going to be our blue and violet. And so they're the ones that have the most energy, and they're going to look like this. Their wavelengths are going to look like this versus, again, our red will look like this, okay? So again, those different wavelengths of light, those photons traveling at different wavelengths are going to stimulate the different receptors, whether it's, again, your red cone or whether or not it's your blue cone. Or whether or not it's your yellow cone, okay? So they're going to stimulate those different cones separately. Now, the color that the eye perceives, okay, is going to be a reflection of that wavelength of light. So, for example, if you see green grass, okay, the reason that you're seeing green grass is that all the other wavelengths of light are being absorbed by the pigments here. And the wavelength that's actually being returned to your eye, reflected and returned to your eye, is this green wavelength because it's stimulating, again, those photoreceptors associated with the color green. So what's interesting is we associate like green with the color photosynthesis, but actually it's the one wavelength of light that's not being used for photosynthesis. And that's why it's reflected away, right? So it's always kind of, I think, interesting that we associate green with photosynthesis, and it's the one wavelength of light it's not using for photosynthesis. So again, it's reflected back to your eye, and that's why you see the green. Now, when you look at something that's white, like this word here, it looks white because all of the wavelengths of light are being reflected back to your eye, stimulating all of the cones. And then black is going to be just the opposite. So this word black looks black because all of the wavelengths of light are being absorbed by the pigments in this word here, and none of the wavelengths of light are being reflected back to your eye. So black is actually the absence of light for you. It's kind of interesting. Now, refraction. Obviously, light, I'm not going to go too much into this because hopefully you already know refraction. Refraction is just the bending of light as it enters different medium. So because, again, our photons are traveling at a certain speed, as they enter a new medium, that's going to change that speed and hence the path of light. And so it usually will kind of send it off at an oblique angle here. So that is what we call refraction. It's just the bending of light as we go from something like air to liquid. Okay, so the lens of the eye is going to refract light, as well as the cornea of the eye is going to refract light as well. So when we look at the lens of the eye is convex. And so you'll see it has this kind of convex shape here, where it's thicker in the center than it is at the edges. And then if something is more concave, because we'll talk about some issues with the shape of the eye, then it's thicker at the edges than it is in the center. Okay, so just two terms, hopefully you already know those. So when we look at, again, the eye and the lens, we have this convex lens that's going to bend the light passing through it so that the rays will kind of converge on these focal points. And what this does, more importantly than kind of knowing that the rays converge at the focal point, is that I want you to see that what it ends up doing is, as the light is refracted, what's up top ends up on bottom, and then vice versa. Okay, and then it's also reversed from left to right. So it's just like when you're looking through a microscope, right? The refraction of that light ends up putting off the image so that the image is formed upside down and reversed. So why is it that if this is the image that's being portrayed on your retina, why is it that you don't see the world upside down? And I'm pretty sure you can probably figure this one out, right? It's your brain. So again, your brain processes this and then flips it back for you, okay? Now, the crazy part is that if you were to, and they did this experiment where they took a bunch of people, they put them in a room, they put these glasses on that refracted the light and flipped the world back upside down and left to right, reversed it for them, and they made them wear them for like 2 weeks. And the crazy part was, by about a week in, their brain had already taken that image and flipped it upright. Okay? It just took a little bit of training. So they wear them like that for another week, right? Then they take the glasses off. And what happens when they take the glasses off? Their world is upside down and reversed. So then it took another week for their brain to adjust and go back to kind of its normal image. So it was just interesting to see how much your brain is involved with your different senses. So again, this is just showing you again that because of the lens of the eye, you end up getting this image portrayed on the retina that is upside down and reversed. Okay. So as light enters the eye, it has to enter the cornea, which is actually refracted by the cornea here, then through the aqueous humor to the lens of the eye where it's refracted again, and then through the vitreous humor, finally to that neural layer of the retina where it can stimulate the photoreceptors. Okay, and so you should be able to kind of, especially for lab, visualize these different layers of the eye as the light enters it. So again, the light is going to be refracted at these 3 points: cornea, okay, on the outer cornea, then the lens, okay, here. And then as it's refracted back out the eye. So the cornea and the lens obviously are going to be the two big refraction points that are going to occur. So you can see you get a little bit of refraction at the cornea and then even more refraction at the lens. Those are the two big refraction points that are going to portray that image on the retina, okay? And the majority of the refractory power is actually in the cornea, but the cornea is constant and doesn't change. So it's the lens that we're able to change. And as we change that lens, right, by contracting those and relaxing those ciliary muscles, and technically, actually, when you I just think this gets more confusing and it's more than you need to know. But technically, when the ciliary muscles contract, they actually loosen these suspensory, because it's circular like this. So it actually loosens these suspensory ligaments and allows the lens to relax. But that's, again, a little more detailed than I want you to know. I just want you to know that the ciliary muscle and is part of the ciliary body, and that it controls the shape of the lens of the eye. And as it puts tension on those suspensory ligaments, then it's going to cause the lens to stretch and flatten out more. And then as it releases that tension by technically contracting, then it's going to allow that lens to bounce back to its convex shape. Okay. So again, if you're focusing for distant vision, if you're trying to look again far away, then you are going to, again, stretch out that lens. Okay. So the ciliary muscles technically relax, but they pull on, when they do, they pull on those suspensory ligaments and stretch the lens. Okay. And so that the big thing is that the lens is stretched when you're looking far away, okay? And so if you, again, can see 20 feet, then you have normal vision, okay? You're normal. So again, the lens is going to flatten out for distance vision. Now, when you're reading up close, that's when the lens is allowed to bounce back into shape. And so, and again, I'm not, you don't have to know sympathetic versus parasympathetic for this. I really just want you at this point, because we're kind of running out of time, I really just want you to know that the lens is stretched when you look far away, distance vision. And then when you're looking up close, the lens bounces back, okay? It's elastic, so it kind of bounces back to its convex shape. And that's what's supposed to allow you to see up close. Now, again, as we get older, because this lens over the span of our lifetime is stretched and back, stretched and bounces back, stretched and bounces back, it starts to lose its elasticity. And as it loses its elasticity, it doesn't bounce back all the way for our close vision. And so it, ends up being a little flatter than it used to be when we were, in our early 20s. And so it makes it more difficult for you to read up close. And that's why as we get older, we typically need reading glasses. So we call that usually presbyopia, because it's something that happens to all of us as we get older, as the lens loses its elasticity. Now, problems with the actual shape of the eye that impact refraction are a couple things. So I am personally what we call myopic. It's nearsighted, which means I can see up close really well without I used to. Now I have some presbyopia with the loss of elasticity of my lens, so bifocals are in my future. But for the most part, I used to be able to see up close just fine. Didn't need any glasses or anything. But my distance vision is terrible. I'm like near blind. And so that's because the shape of my eye is too long, and the focal point ends up being in front of the retina. And so, again, we call it nearsighted, myopia, nearsighted, because you can see up close but not far away. We can correct this with just putting in a more concave lens, either via glasses or a contact lens of the eye. Now, there are some other people that are what's called farsighted or hyperopia. And hyperopia is where your eye is the opposite. It's too short and the focal point is way behind the retina. And so this is corrected with a convex lens. So those are the two kind of big clinical, you know, clinical terms that you would need to know for the shape of the eye. And then astigmatism is just where you have these unequal curvatures of the corneal lens. So these are a little bit more difficult to treat. Laser procedures are ideal for that. So again, this is just showing you a myopic eye that's too long and the focal point is up here. So again, your distance vision is all messed up. And then here, if your eye is too short, then the focal point is behind the retina. Okay, so again, you need a more convex lens to correct that. So those are the big ones that you have to know. All right, so let's get into the last portion of this, and I'll try to turn this. I know this lecture's gone long, but I don't want to cut out the vision section. I want to keep it all together. So the last section is going to be talking about phototransduction. How do we go from that light energy to, again, nerve transmission down that cranial nerve? Or I'm sorry, down the cranial nerve to the optic nerve. So what we're going to talk about here is how we stimulate those photoreceptors, the rods and the cones, and then how they send that information back again to, they're going to send that back to the bipolar cells and then to the ganglion cells. Okay, so that's what we're going to talk about. I'm not going to break this up into the outer segment and the inner segment because, again, this is just making it a little more complicated. Sometimes your book gets too complicated. And so we're just going to talk about the train, basically, of events that have to occur to get us from light coming into the retina and neural transduction. So where we're sending that message back through the nerves. Okay. So when we look at our photoreceptors here, we're going to have our rods and our cones. And again, you're going to have, especially in the periphery of the eye, more rods than you will cones. So let's just do a little comparison of these photoreceptors. So remember, rods are going to be for non-color vision. Cones are for color vision, see color vision, right? And we have 3 of those cones that are going to absorb wavelengths, slightly, again, different wavelengths of that visible light spectrum. And so what we see in terms of color vision is going to be a mix of which of these color receptors are going to be stimulated, okay? So again, you know, if you've stimulated some red and you've stimulated some blue and then they overlap in this one area, you've stimulated both red and blue receptor wavelengths, or photoreceptors with those wavelengths, then you're going to get kind of more of a purplish color, okay? So that's how you get kind of the crossover of all of the different kind of gradations of color between just these 3 different photoreceptors. So again, cones, color vision, rods, non-color. Rods are going to have a high sensitivity. And what that means is that they're going to trip with just a little bit of light. Okay, so they function in dim light. Where cones are the opposite. They have low sensitivity, which means, again, to stimulate them, you need bright light. Okay, so here, dim light, bright light. That kind of makes sense, right? Here, our rods, we're going to have low visual acuity, okay? And so that means that things aren't going to be as clear, okay? They may be a little more fuzzy. And so that's because many rods are going to converge together onto one single ganglion cell, okay? And our cones, we have high visual acuity, especially in that fovea centralis. And in the fovea centralis, we actually have 1 cone per ganglion cell. Okay, so that's why it gives us a sharper image. Our rods, again, are going to be more numerous, and our cones are going to be less numerous, especially, again, on the periphery. And again, mostly in our peripheral retina, and this is going to be mostly in the central retina, again, concentrated especially in that macula lutea and this fovea centralis. Color blindness. So color blindness happens if you lack, again, it's a genetic disease. It's an X-linked condition, meaning it's carried on the X chromosome. So since men only have one X chromosome, it's more common in men because they only have one chance, right? At, not having that color blindness gene. Females have two chances, okay? So because we have the two X's. So again, color blindness means that you're going to lack one or more of those cone pigments. And it's more common in males, again. As many as 8 to 10% of males have some form of it. Again, not the number that I care that you know about, but just kind of interesting. What you really have to know is, again, color blindness is genetic. It's a lack of one or more pigments, more common in men because it's X-linked, okay? And the most common type is red-green, in which either red cones or green cones or both are absent. So you can see this is what stoplights look like for a person that is experiencing red-green color blindness. And again, it just depends on which cone is missing. Sometimes the red can appear green or vice versa, but it just depends on if you're missing one of those cones or both of those cones. And so typically, usually these individuals rely on different shades to give their cues, or, they just know the order in terms of stoplights. Now, I know someone that is actually red-green colorblind. He's like, yeah, I just know the order, so I just go off of the order. So you can test yourself here to see if you can see the path that runs through here. If you can't see that, then you, again, may have red-green color blindness. You probably know if you do. So the last thing we're going to talk about here is, again, how we get from stimulating these photoreceptors is kind of the toughest part. Getting from these photoreceptors, stimulating these to, again, getting nerve conduction down the optic nerve. So we have these photoreceptors, okay? Again, whether you're in a rod or cone, we have these light-absorbing molecules in the membranes of the photoreceptors. And they're called like retinal complexes here. So here you've got this retinal molecule in this other protein called the opsin. And so this retinol molecule combines with the opsin proteins to form 4 different types of visual pigments, okay? And so this is going to be these 4 different types of pigments. And again, it's because they're combining with these 4 different types of opsin. So they all have the retinol, but there's going to be 4 different types of opsins. One opsin for rods, one opsin for your, red cones, one opsin for your yellow cones, one opsin for your blue cones, okay? Or at least those kind of wavelengths. So again, the retinol is going to absorb at these different wavelengths of light depending on the type of opsin that it's bound to, whether or not it's bound to a rod opsin or one of the 3 different cone opsins, okay? So all of these photoreceptors are going to work the same in which I'm going to talk about this. So as I talk about this, we're going to focus on what it looks like in rods, because we're going to talk about bleaching of your rods. But again, know that it's going to work this way in all of these different photoreceptors, rods and cones, all 4 different ones, okay? So what's going to happen is you have, again, this retinol here, okay? And retinol is derived from vitamin A. So that's why your vitamin A is so important, okay? So that vitamin A is going to make up this very important retinol protein. And normally this retinol protein is in what it's called the cis conformation. It's in this kind of bent shape called the cis conformation. And so it's again combined with the opsin to form what we call rhodopsin. Okay, so that's again kind of the beginning. The beginning form is we have this cis retinol form inside the opsin. Okay, now what ends up happening is when light is absorbed by this photoreceptor in the wavelength that it requires, okay, it's going to trigger a change in this retinol to go from the cis conformation to what's called the trans conformation. Here's the trans conformation. So instead of kinked now, it's going to be in this straight conformation here. Okay, so we call that the trans. So it goes, and you can think of, it starts in cis, C comes before T in the alphabet. So it starts in cis. When it absorbs light, it's going to cause it to change to the trans because T comes right after, right? So hopefully that'll help you to remember which comes first and which comes next. So the trans form, okay, here is going to be the kind of active form when light activates it, okay? Now, what ends up happening is here eventually, okay, that trans retinol will be converted back to the cis form by enzymes, basically. Okay, so eventually it'll get transformed back to that cis form, and then it'll be ready to start all over again. Okay. So we'll talk about kind of how this happens. So this is just showing you the same thing here. We have our cis retinol in the opsin form and the rhodopsin. Again, light's going to be absorbed. It's going to undergo a conformational change to that trans form. And then that trans retinal form actually separates a little bit, and that causes the opsin to lose its color. That's why we will say your rods are bleaching. And we'll talk about why that happens differently in rods versus cones. Okay. And then here again, but remember this process happens in your rods and cones right here. Okay. And then when eventually over time, and this time period, okay, that brings us back here is going to be different in your rods versus your cones. But eventually, again, if that's not reactivated by light here, it's going to end up with enzymes bringing it back to its cis form, okay? So that's kind of a regeneration. In darkness, the enzyme converts back to its cis retinol form, okay? And then it's ready to be stimulated again. So let's talk about, again, what happens in the membrane here, okay? And so here's our photoreceptor membrane. Now, in a normal, again, photoreceptor membrane that has not been stimulated by light, what's going to happen is we have these gated ion channels that are open. And they're going to allow sodium and calcium. They're going to allow cations to come into the cell. And this allows the cell to stay in a kind of normal but a normal state, okay? So we have to have this gated ion channel open, okay, to allow some positive ions in, because otherwise just potassium is going to leak out and it's going to hyperpolarize the membrane. So we need this channel in to let some positive ions in, okay? So in the dark, think gated channels open. In the dark, gated channels open, okay? It's a cation gated channel. Now, in the light, what happens, and I'm going to simplify this. You do not have to know all these steps, okay? It's too much. In the light, what's going to happen is, again, the light is going to convert that cis retinol to trans retinol. When it does that, okay, it is going to activate, okay, these G proteins. Remember the G protein cascades, okay? So it's going to activate one of these G protein cascades. And eventually what's going to happen is it'll activate this enzyme at the very end that's going to break down this cyclic GMP that was needed to keep this open, okay? So again, light comes in, converts cis retinol to trans. Trans retinol activates this G protein cascade that eventually breaks down that GMP, that cyclic GMP, and closes this channel, okay? Now this gated channel is closed, and when this gated channel closes, we can't let positive ions in, and the membrane is going to hyperpolarize, okay? So if it hyperpolarizes, now, okay, it's going to stop releasing transmitter. Now, this is the tricky part, okay? So in these photoreceptors, remember, there's no action potentials. It's just all graded potentials, okay? So again, when the light hyperpolarizes those photoreceptor cells, they're going to stop releasing neurotransmitter, right? Because now it's going to have this hyperpolarizing effect, so it won't release neurotransmitter. Now remember, we talked about some receptors are going to release excitatory neurotransmitters, and then we mostly talked about that. But this is a case where that photoreceptor was actually releasing inhibitory neurotransmitter, okay? It was actually releasing glutamate to the bipolar cells. So now, okay, when light hyperpolarizes that photoreceptor cell, and now it stops releasing that inhibitory neurotransmitter, it stops releasing that glutamate to the bipolar cells. Now the bipolar cell is no longer inhibited. And now the bipolar cell can depolarize and release neurotransmitter to the ganglion cell. And then the ganglion cells are where the action potential is generated. That's going to bring that information back to the brain. I know, you're like, whoa, that was a lot. Okay, so let me bring this to your kind of next level here, okay? So let's back up and talk about what happens here, okay? In the dark, remember that this gated channel is open. Remember, in the dark, gated channel's open. It allows, again, for positive ions to come in. So it allows depolarization, no hyperpolarization. So again, here in the dark, channel's open, allows a depolarizing effect, okay? Allows neurotransmitter to be released, which is inhibitory because it releases glutamate, okay? So as that glutamate comes across here, it's going to inhibit the bipolar cell, hyperpolarizes it and inhibits it. So no message gets sent down the ganglion cell, okay? So let me go over this again. In the dark, channel stays open, okay? It allows depolarization. It allows then neurotransmitter to be released, which is glutamate. Glutamate inhibits, it's an inhibitory neurotransmitter, inhibits, again, this bipolar cell and causes hyperpolarization. So again, now this cell is not going to release neurotransmitter, and now the ganglion cell is not going to receive any excitatory potential, okay? So no action potential. So that's in the dark, okay, for all the receptors. Now, all of the receptors, again, whether it's a rod or it's a cone, one of the three cones, depending on what wavelengths of light. So if this is a cone and it's one of the red cones, it has to receive the right wavelength of light, right, to stimulate it. But if it receives that perfect wavelength of light, okay, or that right range of wavelength of light, then it's going to come in, and what's going to happen? The light is going to change the cis-retinol to trans-retinol. The trans-retinol is going to go through that cascade of G protein effect, right? And it is going to shut down this gated channel because remember, it's going to break down that cyclic GMP that it needs. So it's going to shut down this channel. When it shuts down this channel, it's going to hyperpolarize the cell. When it hyperpolarizes this, it's going to now prevent the release of the neurotransmitter. Remember, that was glutamate inhibitory. So now no inhibitory neurotransmitter means, yay, your bipolar cell can now release neurotransmitter because now it will depolarize, okay? because it's no longer hyperpolarized. So now depolarization is going to cause, again, neurotransmitter to be released, then now stimulating an excitatory and then action potential in that ganglion cell. Okay, so these are the two biggies. If you can kind of, again, I'm not saying that I'm going to have you write out these steps, but if you could just recognize these steps, that's what's going to happen in your photoreceptor transduction. This is how you go from light energy to action potential. And remember that these ganglion cell axons are going to make up your optic nerve. Okay, so this is just going to go over those details again. In darkness, the sodium channels are held open and the photoreceptor is always partially depolarized. Okay, so then it's going to continually release inhibitory neurotransmitter onto the bipolarized cells and suppresses the activity. So again, no neurotransmission down that optic nerve. Now in light, the enzymes, again, because of that movement of the retinol from the cis to the transform, okay, is going to cause, again, that closing of the sodium channel because it's causing the breakdown of that cyclic GMP that was needed. It's going to close that sodium channel and produce a hyperpolarized receptor potential, okay? So that will then inhibit the release of inhibitory neurotransmitter, okay? So it inhibits the release of inhibitory neurotransmitter. And then the bipolar cells are excited and send a nerve impulse again to the ganglion cell, down that axon and to the brain, okay? So that's how it all happens, all right? So again, rhodopsin is so sensitive to light that bleaching occurs even in just starlight. So in really bright light, the bleaching occurs so fast that the rods are almost non-functional. That's why if you go again from a dark room to a light room, like for a minute, you're like, I can't even see, like I'm just not functional, right? And we'll talk about why that is in terms of the rods in your eyes and how they adapt differently. So again, cones are the opposite. They need that bright light to be activated. So again, we'll talk about kind of light adaptation and dark adaptation here. So light adaptation, again, is going to happen when we're moving from darkness into bright light. And we're going to see a glare because both our rods and cones are going to be strongly stimulated. So the reason you can see anything, right, is because your cones are stimulated. And you have large amounts of the pigment are being broken down instantaneously, and that kind of produces this like glare for us. And then the pupils will constrict. But over time, visual acuity is going to improve over 5 to 10 minutes. And that's because what's going to happen is your rod systems will be kind of turned off and reset. So again, those rods will eventually kind of turn off and retinol sensitivity will decrease, okay? That requires about 5 to 10 minutes. So that's why it takes about 5 to 10 minutes for you to adjust to the light coming out of a dark room. And the cones and neurons, the cones actually will adapt really rapidly, okay? It's your rods that adapt more slowly. So then dark adaptation is when you're moving from a bright light into darkness. And we see blackness when you walk into a dark room, you can't see anything sometimes. And that's because the cones stop functioning in that low intensity light, right? But the bright light already still has your rods bleached. So kind of like none of your receptors are working, right? So your bleached rods are still turned off. And so again, and this is the reason why your rods contribute very little to your daylight vision, since they're bleached as fast as they can regenerate, then you bleach them out again. So again, that's why, again, it takes a while for you to adjust to that dark room until all your rods are reset, and then you can your rods take over. So again, rhodopsin accumulates in the dark, and so retinal sensitivity starts to increase again as you're in the room longer, in that dark room longer. And So sensitivity increases usually within about 20 to 30 minutes. Full regeneration of your bleached rhodopsin takes actually 30 to 40 minutes in your rods, where it's only 90 seconds to regenerate the cone photopigments. So you can see why there's the difference here. Again, that's why you have such a hard time adapting to a dark room. And that's why it takes you a full 30 minutes to actually be able to be kind of fully adapted to the dark room. Okay. Again, your cones are going to adapt much quicker. And just as a reminder, then again, that information as it's sent back through those ganglion neurons, right? Through the axon of the ganglion neurons that are going to make up the optic nerve. Again, then they'll come back here to the brain through, again, sometimes the optic chiasma if they cross over. And as they enter again, the brain, they'll eventually be sent back here to that primary visual cortex, okay, where that information will be processed. Now, just kind of as a fun thing, your depth perception is where both of your eyes view the same image from slightly different angles. So the visual cortex will actually fuse these slightly different images, kind of giving you that 3D image. And that gives us our actual depth perception. But it does require, again, input from both of our eyes. Again, viewing that same image from slightly different angles. So that's what actually gives us our depth perception. It's kind of fun stuff. Okay, so this is the end of this. I'm sorry this ran a little bit long. Please let me know if you have any questions, and I will try to make this next lecture on the anatomy of the ear a little bit shorter. I'll try to truncate it for you a little bit.